Yeah, you guys. Well, now we're to the third video of chapter 11, part 1. And we're going to visit about, we're going to continue visiting about neuroanatomy. So to begin, I just want to get you guys excited about the topic, um, neuron, and just really give you a little bit of stats on the neuron. Neurons are long-lived. They live over 100 years. So the neurons that you're born with, for the most part, are the ones that you're going to die with. So it's amazing. It's amazing that they live that long. They're amitotic. So mitosis is when a cell divides. So basically, when one cell becomes two cells. And that happens all the time in your body in many, many different cells. However, neural tissue does not divide. So it's a big deal if we damage it. So even though it's really exciting that it's long lived, it's a bummer too because we don't really grow new cells, new nerve cells. They're also very expensive in terms of using sugar and oxygen. So it has a high metabolic rate. So it uses lots of oxygen and glucose. So it's expensive in terms of energy resources. And we'll get to talk about how it uses that energy and so forth in the next couple of times that we get to visit. All right, so let's go over the basic components of the neuron. And I have alluded to it a little bit, but we really haven't gone over it, in together. So let's do that. So let's begin. So here's our neuron. So let's start with the dendrites. So all of these little projections are called dendrites. And I believe dendrite means branch. Okay, so here's our little dendrite, many different dendrites. Then we have the soma, which is the cell body. Remember the somatic senses? That means body as well. So here's our soma. There's the nuclei of our cell. Then we're going to move to the axon hillock. And that's really important when we get enough information via these dendrites and other nerves talking to this soma. If we get enough information that says, yes, neuron, I want you to fire, we have to be able to have that information be strong enough, and I'll talk about what that really means in the next chapter. That information needs to be strong enough to get past the axon hillock, which is right here. So once that information is past the axon hillock, it's an all or nothing deal. So if we get past the axon hillock, that information is going to be sent. It's like pressing send, thank you, pressing send on an email. Once it is gone, it is gone. So the axon hillock is that send button on an email. Okay, it's all or nothing. Once it's gone, it's gone. Okay, and then we have the myelin sheath. So here's the myelin sheath here. Here's some more. Here's some additional pieces of myelin sheath. So it's that fatty insulator that I talked about. And then in between these little fatty sheaths, these myelin sheaths, are spaces where there's no myelin. And we call those nodes of Ranvier. And that's a pretty fancy name. And it's probably a fancy scientist who discovered it. So I don't really have a great way of remembering that. It's kind of a fun thing to say, though. So you could try that, node of Ranvier. And so really the information, in order for the conduction velocity to happen quickly, the information actually, the electrical current essentially, bounces over the top of that cholesterol sheath, that myelin sheath, and then pops in at the node of Ranvier, gets more energy, and then goes over the myelin sheath till we get to the next node of Ranvier, gets more energy, then pops back over. So you can see how that myelin sheath is gonna speed everything up, right? Because the action potential doesn't have to go through the whole axon. So, and we'll talk about that more. But it permits for this. What it does is permit this saltatory conduction. So this hopping of the electrical current. Okay, and I have this mental picture. I'm trying to think of how to remember saltatory conduction. I have this mental picture of pouring salt in a hot pan and watching it bounce around. And I honestly don't even know if that's true. If you did that, if it would really happen. but that's just how I remember it. So if anybody else has another fun way, go ahead and, in terms of remembering what saltatory conduction is, post it on tips, because I'm always looking for new ways to help students learn, and try to attach new information to old information. So do share anything, any silly thing. I share mine, and I guarantee you guys think some of them are pretty silly. Um, but yeah, so saltatory conduction. Uh, and then we'll go ahead and talk about gray matter and white matter. So white matter are these axons that are covered in myelin. Okay. And you want to think, so any axon, think of it like a telephone cord. It's not, and maybe the cell body and the dendrites as the person at one end of the telephone cord needing to send that information. So integration of whatever information is coming from other neurons or other parts of the nervous system is happening here, just like a person is thinking, deciding, formulating language in terms of what they want to say. So this is where really, the thought per se happens here. Okay. So at the end of the phone cord. Now, if we're going to send that information, it's got to travel through the phone cord, okay, or the information highway. Okay. And then I talked about the gray matter, which is the cell body and the dendrites, as being integration of the information or the city. So that was a different analogy I used. So we had the city here. So everybody's working together, getting stuff done, and then we need to get home, right? So we're going to take the information highway home. Not a lot of thought here, none at all actually. Just sending information that's already been determined here. Okay. All right. And then, so here's our axon. Let's travel down our axon, and we're going to get to the teleodendria, which are just the end parts of the axon. And then at the very, very end of an axon are going to be these terminal end buttons. And they contain these things called synaptic vesicles. And when we look at pre and post synapses, I'll show you those synaptic vesicles. But they house neurotransmitters. And neurotransmitters are these chemicals that we use to relay this electrical energy from this axon. We turn it into a chemical energy, which is those neurotransmitters, which then elicit a new electrical chemical response in the next neuron. So we'll talk about that in the next week or so. All right. So now, here's our neurotransmitters. Okay, so we had talked about synaptic vesicles at the terminal end button. So here's a terminal end button. You can also, so inside those synaptic vesicles, which are depicted here in green, these little circles with the dots, okay, there are neurotransmitters, and they communicate with the next neuron. So what happens is this axon, so this is the very end of the axon. So let me go back here. So here's the terminal end button, and we're going to blow it up, okay? So the information has been sent here, and we're going to go to the next page. So this is a terminal end button blown up. So information is traveling this way, and the synaptic vesicles are then told to release this neurotransmitter, okay? And they do so into the synaptic cleft, which is basically, here's another neuron. So this is the presynaptic neuron, this is the postsynaptic neuron, and there's a little lock and key function where the neurotransmitter fits into this little lock, and then kind of opens up a passageway to enter the postsynaptic neuron, okay, and send that information. All right, and the synaptic cleft is the space between the pre and postsynaptic neuron. Okay, and we always think our typical mental process is to think that when we ask a neuron, when we send an action potential, we're always sending an action potential, which is that information that we want, the information from one neuron to the other. And we're always thinking, we're trying to tell that neuron, another neuron, that it needs to fire, but that's not always true. We're doing a lot of inhibition of neurons as well. So we both have excitation and inhibition of neurons, and it's happening all of the time. So just as much, if not more, inhibition as there is excitation of neurons. And then we have mitochondria, which you guys might already know are my favorite organelle. So there's these little guys here. They look like jelly beans with squiggles inside. And they're really responsible for uh, creating the energy needed for this process to work. So they take in sugar and oxygen and turn it into adenosine triphosphate, which is the cash currency of energy in our cell. And we get to learn about that later. Just warming you up for it. And I don't know if you guys remember, but I talked about how energetically expensive this system is. Um, and in order to use energy, we need to have mitochondria on board. So we have tons of mitochondria in our nervous system. Here's more. They're all over. So you're using tons of mitochondria. You have tons of mitochondria in your nervous system because you need lots and lots of energy. But you have mitochondria in other cells too, your muscle cells, for example. Uh, so so super cool. Gotta love the mitochondria. All right, now we're going to talk about our glial cells. Remember, the unsung heroes of our nervous system. So we just spent a little bit of time on the neuron. Now we're going to talk about the glial cells. So we have 5. So astrocytes are the most versatile. Think of them as being, I don't know, I think like astronaut because they look kind of star shaped. So it's these guys right here. And that's sort of how I remember. And they have these projections. So they anchor a lot of the blood vessels and neurons where they need to be. Okay. And they also tell young neurons, they help send young neurons make the right connections. So isn't that cool? It helps guide a new neuron during development to make the right connection with another neuron. So it is just a cool thing, right? It also is a bit of a janitor. So it mops up leaked potassium, and we'll talk about why that's important in a little while. So don't get too hung up on it. Um, we use potassium to send those signals, those action potentials, or that information to the neuron. And sometimes, and so we transfer potassium back and forth across the cell membrane, in order to do that. And sometimes we get too much potassium out here, so we have the astrocytes come clean it up, which is super nice. And they also recapture, release neurotransmitters, and you guys might be able to speculate on why that would be important. So if we have a bunch of extra neurotransmitters kind of flitting around in the interstitial fluid, and interstitial fluid is the fluid between the cells, then that neurotransmitter could then inadvertently unlock another neuron and tell it either to be inhibited or to fire, right? When we didn't want it to. So we really don't want a bunch of neurotransmitters just off willy-nilly, right? So we have the astrocyte cleaning that up, which is super important. And it also helps form the blood-brain barrier. So the blood-brain barrier is an important thing. So blood on your neural tissue is a really bad thing. It can cause it's kind of like throwing gasoline on a fire. It causes a lot of excitation of nerves inadvertently. Um, so we actually don't have blood washing over our neural tissue. We have cerebral spinal fluid. And we get the nutrients from our blood, and we do need blood to go to our brain, but the nutrients is then kind of put into the cerebral spinal fluid, and then we feed our neural tissue that way. So, so it helps form that blood-brain barrier so we don't get blood into onto our neural tissue. So if you have a patient that's had a hemorrhagic stroke and they get blood all over their neural tissue, it's a really bad thing. It can cause a lot of damage. All right, now we get to talk about the microglia and the ependymal cells. So I always think of the microglia as the little nurse cells. They're the ones checking in to make sure that the neuron is healthy. Okay, and it also cleans up messes just like a nurse might, and so it phagocytes or eats microorganisms and neural debris. Okay, so any foreign body that might be in or around that nervous tissue. And then we get to talk about the ependymal cells, which I think are so pretty, and they have such a pretty name. It's actually called wrapping garments, and they line the ventricles in the brain. And we're going to get to them in just a minute, so don't worry yet. But basically, the ventricles house the cerebrospinal fluid. But so basically, we have these ventricles that are in the brain that are housing the cerebrospinal fluid. But we need that cerebrospinal fluid to flow, right? Kind of bathe the tissue, and we need the cerebrospinal fluid to be able to penetrate the through the ependymal cells to get to the neural tissue, right? So we have these wrapping garment cells, these ependymal cells. So they form a permeable barrier where the cerebrospinal fluid can flow and make contact with the neural tissue, and it also helps circulate. Just like, remember that PCCET, that pseudostratified ciliated columnar epithelial tissue? Remember that cilia on it that help move, foreign debris out of the respiratory tract? Think of little cilia in your ventricles in your brain, and we're going to learn about those in just a minute. Moving that cerebrospinal fluid around, just kind of washing it through. So here's an example of the ependymal here. So they kind of honestly look like epithelial tissue. They have the basal and apical edge like an epithelial tissue. So if that helps you remember ependymal and epithelial, if you pair those, that might be a good way to remember it. All right, now we get to talk about the oligodendrocytes and the Schwann cells. They have essentially the same jobs, except for one is in the central nervous system and one is in the peripheral nervous system. So oligodendrocytes are in the central nervous system, and they form myelin sheath. Okay, and Schwann cells do the same thing, but in the peripheral nervous system. So I think of the Schwann man driving around the periphery, right, in our neighborhood, selling ice cream. And ice cream kind of looks like a white fatty sheet. So I think the Schwann man is in my peripheral nervous system. So I hope that helps you guys. So we have oligodendrocytes in the central nervous system, and then the Schwann man selling ice cream and making myelin sheaths in the peripheral nervous system. Okay, so our Schwann cells. All right. Now I did talk about this picture a little bit in terms of the pre and postsynaptic neurons. So this is the presynaptic neuron. This is the synapse here. Here's a synaptic cleft, and they're making a synapse. And then we have the postsynaptic neuron. All right, so we've talked about motor neurons and sensory neurons a little bit. Motor neurons are always efferent. They activate muscles or glands. And then we differentiate them based on size, how fast they send information, which is conduction velocity, and the amount of myelination that they have. So in general, the wider and more myelinated the axon, the greater the conduction velocity, the quicker the information is going to be sent. Okay? All right, and the sensory neurons are always afferent. So motor is efferent, sensory is afferent, and then interneurons are neurons that don't target a muscle, for example. So we talk a lot about sensory neurons and motor neurons because they have a target. Okay, they're sort of the last line in the loop, but there are many, many more interneurons making that connection between them. Okay, so they make up the largest number of neurons in the brain. They do not exit the central nervous system, so they're only in the brain and spinal cord, and they provide communication between neurons. They look essentially the same. Okay. All right. So then we have different types of synapses, or different ways that neurons can talk to each other. So the first one is axodendritic. So just break the word up. So we have an axon articulating or communicating with a dendrite. So here's axodendritic here. Here's one here. We have axosomatic, so axon and soma. Axoaxonic, and so here's one here. And then less common ones are somasomatic, and we don't have a picture, but you can imagine two neural cell bodies communicating directly without the help of an axon or dendrite, and then two dendrites communicating, which are dendrodendritic synapses. Now we get to talk about cerebrospinal fluid, specifically the ventricles and the meninges. So the cerebrospinal fluid, it cushions the brain and spinal cord, so it essentially helps alleviate 97% of the brain's weight just by basically suspending it in fluid. And it also distributes nutrients to the brain and spinal cord and is renewed every 8 hours. So 3 times a day we're getting new cerebrospinal fluid. And it's produced in the choroid plexus, which is located in the ventricles. And here are your ventricles. And when you guys watch the neurodissection video, you'll see the choroid plexus, but they're kind of little gray things. Let me see if there's not a good picture here, but let me see if I can draw one for you. So the choroid plexus would be like right here so that's in the lateral ventricle. And there's some in here. Oops. You'd also find it. Oops. Some there. Okay. All right. So I want to talk to you guys about the flow of the cerebrospinal fluid. So this is anterior. This is posterior. So here are our lateral ventricles. This one and this one. They kind of look like two sheep's horns. Okay. So this is a sagittal view. It's not mid-sagittal. So if it was mid-sagittal, we would only be able to see one lateral ventricle. There are 4 ventricles. However, we have 2 laterals, then we have a third and a fourth. Okay, so just think of the lateral ventricles as being ventricle 1 and 2. And remember, they look like sheep's horns. Okay, so these are our lateral ventricles. And in terms of talking about the flow, we have choroid plexus in here creating the cerebrospinal fluid, also in here. But let's just talk about it from the lateral ventricles. So we have cerebrospinal fluid in the lateral ventricles, and it's going to travel through the interventricular foramen right here and enter the third ventricle here. And then if we didn't have the horn of the lateral ventricle, you would see the cerebral aqueduct right here. Okay, and then the fourth ventricle. All right, and then if we move down just a little bit, you're going to see the lateral apertures and the medial aperture. So here's the medial aperture here, and it's going to send. Oh, and then we have the central canal for the spinal cord. So you're going to have some cerebrospinal fluid travel directly down, and then you're going to have some cerebrospinal fluid travel around the outside of the spinal cord. And then we have our lateral and our medial apertures that send cerebrospinal fluid around the outside of the brain. Okay, and then through the central canal. So the lateral aperture is also called the foramen of Luschka, and the medial aperture is also called the foramen of McGindy. And there's not a great picture of it, but the terms I think you can make sense for the most part. And so through the outside, the lateral or the superficial aspects of the brain where the cerebrospinal fluid is flowing, that space is called the subarachnoid space. Okay, and then the central canal is the most medial part of the spinal cord where the cerebrospinal fluid flows. And so let me see if we get to talk about the meninges. Yes, good. So that's a nice segue in terms of talking about the subarachnoid space. So let's go and talk about the meninges. So we have 3 layers of the meninges: pia, arachnoid, and dura. So it's the pia mater, we call it arachna mater or arachnoid mater. You can call it each, and the dura mater. And mater means mother, and pia means gentle. So we have gentle mother. Then we have arachnoid, which is spider. So it looks kind of like spider web, actually, in the arachnoid space. And then dura means tough mother. So the dura mater is the most superficial covering around the brain. It's actually a thicker connective tissue, like an aponeurosis. It's really a thin sheet of connective tissue. Okay, so here would be Here's the dura mater, and then this blue in here would be the arachna mater, and then the pia mater is right here. So pia mater, oh sorry, arachna mater, and then dura mater. All right, and then let's talk a little bit more. So there's some extensions of the dura. So remember, dura is the most superficial, then arachnoid, then pia. Just think that you want the gentle mother closest to the brain. Then the arachnoid mater is in the subarachnoid space is where the cerebrospinal fluid flows, and the dura mater is the tough mother, and you want that on the outside, right? All right, so then the dura mater has a couple extensions. They're called denticulate ligaments that help hold the spinal cord in place. So this is a picture of the spinal cord where we've pulled away the dura mater, which is right here. Okay. And then we have part of the dura mater connecting to the spinal cord. So it keeps the spinal cord in place. All right, and then we also have two additional meningeal structures, the falx cerebri and the falx cerebelli. Okay, so this is anterior, this is posterior, this is the falx cerebri, and it's really thick connective tissue, and this is actually you would find this separating the two hemispheres of the brain, so the right and left. Okay, and then this actually travels right underneath the occipital lobe and then right over the top of the cerebellum, okay, the falx cerebri. So it helps separate and actually protect the cerebellum from having the cerebrum kind of compress or squish it. Okay, it holds the cerebrum up away from the cerebellum here. So here's a cadaver picture of the falx cerebri. Okay. So this is all the falx cerebri. So on the other side over here, that's a lobe of the brain. And then it looks like the left lobe of the brain has been cut away here. All right, so now we're going to talk about the cerebrum and the layers of the cerebrum. So we have pyramidal cells and non-pyramidal cells. So this is a cross section of the cerebrum. This is in the cerebrum. Let me go back up here. So here's the cerebrum here, right? This is all cerebrum. Think telencephalon. So we have pyramidal and non-pyramidal cells. So this is the most superficial. This is the deepest part. Okay. So pyramidal cells are motor. They have motor function, whereas non-pyramidal cells have sensory function, and it's kind of layered like an ice cream sundae. So we have our non-pyramidal cells here. Then we have our pyramidal, which are bigger. Can you see that? Whereas the non-pyramidal are kind of star-shaped, stellate-shaped. So, and then we have the star-shaped again, which are sensory, then motor. Okay, so that's in general all you really need to know for this course in terms of how the cerebrum is set up. Just this layering of sensory, motor, and then you get down to this white matter, which is those myelinated axons. All right, now we get to talk a little bit about the cerebral hemispheres, but first we need to break down a little bit of terminology. So we talk about sulci, fissures, and gyri. So a fissure is a very big sulci. So this is a longitudinal fissure. Okay? So think of sulci as the valleys, and then the gyri are the hills. Okay? So here, these are all gyri, these are all sulci, and then we have fissures, which are really large valleys here. So here's our longitudinal fissure. Okay. And remember, the top of the brain is dorsal, which is opposite. I mean, typically we think of that as superior, right? However, when we're talking about the brain separate from the body, or the brain in general, the top is superior then the bottom is sorry, not superior, is dorsal, and then the bottom is ventral. And then the front is anterior and the back is posterior. Okay? So here's some general landmarks. We have the longitudinal fissure. So remember, it's a very, very large valley, large sulci, which is a fissure. And then we have the central sulcus. And if you think back to learning about the bones, we'll do a brief overview of the lobes of the brain. We have the frontal lobe, just remember the frontal bone, the parietal lobe, which is just underneath the parietal bone, the temporal lobe, which is just underneath the temporal bone, and the occipital lobe, which is just underneath the occipital bone. And so we have a sulci called the central sulcus that separates the frontal lobe from the parietal and temporal lobe. Then we have a lateral sulci that separates the temporal and the parietal lobes. And then we have a parieto-occipital sulcus, which does exactly what it says. It separates the parietal lobe and the occipital lobe. Then we have a transverse cerebral sulcus, which separates the cerebellum, which is I just drew here in blue, and the cerebrum. All right, so what is the function of the cerebrum? The cerebrum, which is the whole brain structure here, interprets sensory information, controls voluntary and skilled muscle activity, and provides emotional and intellectual processing. Right, so here's another view of our picture of our brain. And so this is anterior, posterior, dorsal, ventral. So frontal lobe, parietal lobe, occipital lobe, temporal lobe, cerebellum, pons. Then this is our brainstem, right? Our myencephalon, our medulla oblongata, and so we call it pons and medulla if you'd like, and then our spinal cord. All right, so now I've switched it on you guys. This is anterior, this is posterior. We're going to talk about some general functions of the brain based on location. So humans in general need and want to categorize things in order to make sense of it. And so we've really tried to categorize functions within the brain. So although we're going to talk about discrete portions of the cerebrum as having specific functions, remember, do keep in mind that it really is a huge network and cascade of effects that allow certain functions to work. So even though we talk about speech being in one part of the brain or reading being in another part of the brain, in general, it's not going to happen in an isolated way. Okay? But in order to learn it, we have to categorize it. So in the frontal lobe, we have speech and we have motor. Okay? And we also have intellect up here. So speech, and then we have the primary motor cortex, which is motor control here. So this is just a nice general overview. Then we cross over the central sulcus. This is all parietal lobe. So if you think of motor, speech, and intellect up here, you're doing pretty well. You can have and smell as well. Okay. And then you can think of sensory, body sensory. So somatosensory. Remember, soma is body. So touch and pressure here at the primary somatosensory cortex. And then we have language, which is part of the temporal lobe, and actually is incorporated here with the vision and with occipital and parietal. Okay, but primarily understanding language is in the temporal lobe. We also have hearing in the temporal lobe, and then for vision, we're in the occipital lobe, which is so I think the occipital lobe. So the occipital lobe is in the back of your head, and I'm sure you guys have all heard how your mother or you tell your kids that you have eyes in the back of your head, and so that's how I remember that vision is processed in the back of our brain, which is in our occipital lobe. All right, so this is just one more picture. Um, excuse me. So here's our central sulcus. Another name for that is the Rolandic sulcus. Um, and so you can see that we have speech here, we have speech here. Um, it's really this huge network. But we're going to talk about motor planning for speech here. Okay. So there's one lobe that we haven't really talked about. So we talked about frontal, parietal, temporal, and occipital. We really haven't talked about the insula. So insula means island, and it's actually buried underneath the temporal lobe. So if you resected the temporal lobe, you'd see um, this additional little lobe, and it's responsible for emotional control and homeostatic control. There's a little hidden island there. All right, so let's focus on the frontal lobe. So we're going to talk about the primary motor cortex here in red, also called the precentral gyrus. It's responsible for initiation of voluntary motor movement or motor execution. So that's you deciding to pick up a spoon, it's you deciding to shoot a basketball. It all needs to happen there. And then we have the premotor cortex, which is the motor planning. It also regulates posture, but it's the planning of telling those muscles when and how to fire. And then it's sent up to, so the information, which is here in blue. So the information here, you plan, you create the motor plan, then you set it to the primary motor cortex, and the primary motor cortex sends it down through the corona radiata, which is coming up later, down to your spinal cord, and then out to your spinal nerves, and then to the actual target, which is the muscle. Um, so Broca's area is actually a part of the premotor cortex, and so if we have a patient that's had a lesion to Broca's, maybe from a stroke, they may be able to understand language because this part of their brain is intact. But when they want to produce language, they don't have the ability to plan the motor function. They can't plan the sequence of events that need to happen. Even if the primary motor cortex is intact, the primary motor cortex isn't getting the right information. So it's not, the patient is not going to be able to produce fluent speech. All right, now let's talk a little bit about the parietal lobe. And I try to give you guys just a couple landmarks per lobe to get you oriented. You do get a whole class in neuro, which is super exciting. I just like to give you a nice foundation for when you enter that course. So we have the postcentral gyrus, which is this gyri right here. It's the primary site for sensory input and reception of that sensation from the body, that touch and pressure. Okay. And then we have the inferior idle lobule. It's important for integrating many different senses, so special senses and the somatic sense, so vision, audition, and senses from the body. And then we have the angular gyrus, which plays a role in reading comprehension. So it's this guy back there. So it's actually really close to this visual cortex, so if that helps you remember. All right, so then we're moving to our happy temporal lobe. We have Heschel's gyrus, which is right here. And that's important for processing auditory stimulation or hearing. You might have looked back at other pictures of the brain, so this is where hearing is. And then we have Wernicke's, which is typically on the left side of most patients. Same thing with Broca's, which is that motor planning, that part of the premotor cortex that helps with speech motor planning. Most people are left dominant, meaning that part of their brain is only on that part of their brain is either on the left or the right side, and most people have it on the left side. So same thing with Wernicke's. So it helps you to understand spoken language, the symbolic nature of language. So if you have a patient that has a lesion at the level of Wernicke's, you may find that They can speak fluently, maybe even fluent words, count, or they're going to talk in gibberish and you don't understand a thing they're saying, but they're pretty sure they know exactly what they're saying. And that has to do with a lesion here. So they can't understand speech well, and they can't regulate their own speech because they can't listen to what they're saying and understand it well enough to know if they're saying it correctly. All right, so we've talked a little bit about Wernicke's and Broca's now. There is a connection between the two, a really important one, called the arcuate fasciculus. And fasciculi is just another fancy word for a neural tract. And remember, a tract is just an aggregation of those axonal axons, or just a grouping of axons, right? And so we have a grouping of axons that travel from here to here. So I just think it looks like it's in an arc, and it's a fasciculus, which is just a group of those axons. Okay, so we need information to travel from Wernicke's, understanding what's heard. Okay, then we need to create a motor plan here at Broca's, and then send that information up to the primary motor cortex, and then out to be spoken. So you'll see the loop here. So hearing language, hearing and understanding, sending the information to create a motor plan here, premotor cortex, up to primary motor cortex, and then processed into speech that is then heard by the listener, and vice versa. And then the cycle begins again. So cool! It's a big part of our field, you guys. So it's pretty exciting. And then we have our primary visual cortex back here. This is in our occipital lobe, and that's really all you'll need to know about the occipital lobe for this. So there's one more video I'd like you to watch, and this just gives you a nice general function overview. All right, thank you guys, and I hope you're enjoying this part of the class. I just think it's just a really fun, exciting part. All right, goodbye.